Hello guys and welcome to my channel The Bio Guru, where science becomes sick. And in today's video I will be talking to you all about the Krebs cycle discovered by Hans Krebs. Now before diving into today's lesson, I would like to tell you all that since the Krebs cycle is directly linked to the previous stages of respiration such as the link reaction and glycolysis, it is advisable that you thoroughly understand those topics before proceeding with today's video. And in order to do that, you can check out my previous videos on those topics. With that being said, let's move on with today's lesson, which is the Krebs cycle. So the Krebs cycle starts off when the 2-carbon acetyl group, which was produced as a result of the link reaction, binds to a 4-carbon compound which we call oxaloacetate. So when the 2-carbon acetyl group binds to the 4-carbon oxaloacetate molecule, it forms a 6-carbon molecule which we call as citrate or citric acid, because 2-carbon and 4-carbon gives 6-carbon. So to repeat what I just said, the 2-carbon acetyl group binds to the 4-carbon oxaloacetate molecule, forming the 6-carbon citrate molecule. Now this 6-carbon citrate molecule undergoes dehydrogenation and decarboxylation to form 5-carbon and 4-carbon intermediate compounds. Now at CIE A2 level, you are not expected to know the specific terminology or the names of these intermediate compounds. You just have to know that there are 5-carbon and 4-carbon intermediate compounds formed when citrate is dehydrogenated and decarboxylated. So in my previous videos, I discussed the meanings of the terms dehydrogenation and decarboxylation, but I will talk to you all about them once again. So dehydrogenation, as the name suggests, is the removal or the elimination of hydrogen. And when hydrogen is removed and released, the released hydrogen cannot exist on its own. Therefore, it is accepted by two coenzymes. Now in all the processes which we learned before, that is glycolysis and the link reaction, the hydrogen released was captured by only one coenzyme, but the difference is that in the Krebs cycle there are two coenzymes involved in the accepting of hydrogen. So the first coenzyme is the one that you are already familiar with, that is NAD. And as I said before, according to chemistry, whenever a compound gains hydrogen, it becomes reduced. And therefore, when NAD gains hydrogen, it becomes reduced and forms reduced NAD. Now, the second coenzyme is new to you all, and it is called FAD. You do not need to know what the abbreviations NAD and FAD stand for. You just need to know that they are coenzymes at CIE A2 level. So the FAD coenzyme gains hydrogen to form reduced FAD. So to repeat what I just said, dehydrogenation of 6-carbon citrate occurs to release hydrogen, which cannot exist on its own and is therefore accepted by two coenzymes, namely NAD and FAD, to form reduced NAD and reduced FAD respectively. So now that you are clear with the dehydrogenation of citrate, let's talk about the decarboxylation of citrate. So decarboxylation is the removal or the elimination of carbon from a compound, and in this case carbon is removed from the 6-carbon citrate molecule, because from 6-carbon citrate to form 5-carbon and 4-carbon intermediate compounds, you have to remove 1 carbon atom and 2 carbon atoms respectively. So basically the removal of those carbon atoms is what we call decarboxylation. And during decarboxylation, the released carbon or the removed carbon forms the compound carbon dioxide. So to repeat what I just said, the 6-carbon citrate molecule is decarboxylated to form 5-carbon and 4-carbon intermediate compounds, during which the released or the removed or the eliminated carbon, which cannot exist on its own, forms the compound carbon dioxide. Now, during the conversion of 6-carbon citrate to 5-carbon and 4-carbon intermediate compounds, along with dehydrogenation and decarboxylation, one other important process occurs, and that is the production of ATP or adenosine triphosphate. Now, in the Krebs cycle, ATP is produced by a process called substrate-linked phosphorylation. Now, you should be familiar with the term substrate-linked phosphorylation because I have explained its meaning in detail in my video on glycolysis, so please check that out for more information. However, I will repeat the definition of the term substrate-linked phosphorylation for convenience. So, substrate-linked phosphorylation is the addition of a phosphate group to ADP or adenosine diphosphate to form ATP or adenosine triphosphate using the energy released from the splitting of a substrate or the breaking of bonds within a substrate. So in that manner, ATP is synthesized during the Krebs cycle. So finally, after all these processes have occurred, that is to say after dehydrogenation of citrate, decarboxylation of citrate and ATP synthesis has occurred, then from these 5-carbon and 4-carbon intermediate compounds, the 4-carbon compound oxaloacetate is regenerated via a series of enzyme catalyzed intermediate reactions. You do not need to know what these reactions are, you just need to know that the 4-carbon oxaloacetate compound is regenerated from these 5-carbon and 4-carbon intermediate compounds via a series of many enzyme catalyzed intermediate reactions. So now that you are clear on how the Krebs cycle works, you also need to know the amounts of the different compounds produced during the Krebs cycle. So let's take a look at that now. So these are the 4 main compounds produced during the Krebs cycle, that is reduced NAD, reduced FAD, ATP and carbon dioxide. So let's take a look at reduced NAD first. So during the Krebs cycle, for each acetyl group or per acetyl group, 3 reduced NAD molecules are formed. During the Krebs cycle, per acetyl group, 3 reduced NAD molecules are formed. Now, using your knowledge from the link reaction, you should know that one pyruvate molecule is converted to one acetyl group during the link reaction. Therefore, since one pyruvate molecule gives one acetyl group, the number of reduced NAD molecules produced per acetyl group and per pyruvate molecule should be the same. And therefore, per pyruvate molecule, 3 reduced NAD molecules are formed. So, using your knowledge from glycolysis, you should know that one 6-carbon glucose molecule gives rise to 2 3-carbon pyruvate molecules. One glucose molecule gives rise to 2 pyruvate molecules. So whatever the amount is that is produced per pyruvate molecule should be multiplied by 2 in order to calculate the quantity produced per glucose molecule. And since 3 reduced NAD molecules are formed from one pyruvate molecule, per glucose molecule 3 times 2, which is 6, and therefore 6 NADH or reduced NAD molecules are formed per glucose molecule. So now let's take a look at how much of the other molecules is produced. So for reduced FAD, per acetyl group, one reduced FAD molecule is formed. And since one pyruvate molecule gives one acetyl group, then per pyruvate molecule, one reduced FAD is formed as well. And since one glucose molecule gives rise to 2 pyruvate molecules, as a result of that, per glucose molecules we get 1 times 2, which is equal to 2 reduced FAD molecules. Next is ATP. So per acetyl group, 1 ATP molecule is produced by substrate-linked phosphorylation, and therefore per pyruvate molecule, 1 ATP is produced as well. Therefore, per glucose molecule, we get 1 times 2, which is 2 ATP molecules. I'm not repeating the same explanation over and over again. It's just the same principle which I explained in detail while calculating the amount of reduced NAD produced. And finally, when looking at the amount of carbon dioxide produced, per acetyl group, there is 1 carbon dioxide molecule produced. And as a result of that, per pyruvate molecule, there is 1 carbon dioxide produced. And finally, per glucose molecule, there is 1 times 2, which is 2 carbon dioxide molecules being produced. So the actual thing that you need to remember or memorize is the amount produced per acetyl group. You just need to memorize only that. And from then onwards, if you understand the actual principle and if you have a thorough understanding about glycolysis and the link reaction, which you can get from my previous videos on those topics, then you can use that principle to calculate the final amount of the molecule formed for each glucose molecule, because that is what is needed most of the time in your exam papers. So that's it for today, guys. Hope you all got a good understanding about the Krebs cycle, and if you did, make sure to like, share and subscribe and drop your comments down in the comment section. Thank you all for watching this video and see you soon in another lesson. Bye.